The broadcast is now starting. All attendees are in listen-only mode.
Hi, everyone. Thank you for joining the sixth webinar in our series on the use of NAMs in risk assessment. This series is being co-organized by the EPA, the Physicians Committee for Responsible Medicine, and the PETA International Science Consortium. My name is Amy Klippinger. I direct the PETA International Science Consortium, and I'll be co-moderating today's webinar with Christy Sullivan, who is the Vice President of Research Policy at the Physicians Committee for Responsible Medicine. We have three speakers today who will be presenting on the use of non-animal skin sensitization test methods. First, we'll hear a 15-minute or so presentation from Louis or Gino Scarano and James Cox of the Environmental Protection Agency Office of Pollution Prevention and Toxics. And then we'll hear from Hans Raabe of the Institute for In Vitro Sciences. Before I introduce our first speaker, I'd like to point out that the slides from today's webinar can be found online at the link shown on the screen. And a recording of today's webinar will be posted there as well shortly after the webinar. Slides and recording of the first 5 webinars in this series are also available at this link. There will be time for questions for both speakers at the end of the second presentation. Everyone is on mute, but you can type any questions in the question section within your GoToWebinar toolbar, which you should see on the right-hand side of your screen. Speaking first today will be Dr. Gino Scarano and James Cox. Gino has been a toxicologist with the US Environmental Protection Agency since 1998. While at the EPA, Gino has worked in the Office of Pollution Prevention and Toxics, as well as the Office of Pesticide Program. Prior to joining the EPA, he spent 12 years working in hazard and risk assessment in the public, private, and nonprofit sectors. James Cox has been a biologist for the EPA since 2018. While at EPA, he has worked in the Office of Pollution Prevention and Toxics as a human health risk assessor for new chemical submissions. Prior to joining EPA, James worked as a biologist for the US Army Public Health Center at Aberdeen Proving Ground in Maryland from 2015 to 2018. Before that, he did research in an academic lab at the University of Maryland School of Medicine. And with that, I will turn it over to you, Gino and James.
Thank you. So good morning, everyone. I'd like to thank the organizers for inviting us to give the talk. And as Amy said, my name is Louis or Gino Scarano from the Risk Assessment Division in the Office of Pollution Prevention and Toxics. And with my colleague, James Cox, we're happy to be here. We're going to be providing a brief update on the skin sensitization non-animal methods that we have received in the TSCA New Chemicals Program since the passage of the Frank R. Lautenberg Chemical Safety for the 21st Century Act, which was passed in mid-2016. And the appropriate disclaimer is on our first slide. May I have the next slide, please, Amy? Thank you. So this is what we plan to cover in the next 10 or 15 minutes. I will spend a few minutes talking about the interim draft skin sensitization policy, which we published in April of 2018. And then James will present an overview of the NAM, or New Approach Methods, information that we've received over the past 4 years. And then we will close with the steps we plan to take towards finalizing the policy. Next slide, please. So here in slide 3 is a little background on the draft policy, and the title is here in the red, and then that's the link to go directly to the policy. It was released in April of 2018 and is based on the adverse outcome pathway from initial exposure of a chemical to the development of an adverse event, or the adverse event. In this case, we're talking about skin sensitization. There are multiple non-animal testing strategies in silico, in chemico, and in vitro that demonstrate comparable or superior performance to one of the most widely used lab animal studies for this endpoint, the local lymph node assay in mice, or LLNA. This draft policy is a result of a large collaborative effort with the National Toxicology Program Interagency Center for the Evaluation of Alternative Toxicological Methods, or NICETM, the Interagency Coordinating Committee on the Validation of Alternative Test Methods, or ICVAM, and various international collaborators, and of course, most importantly, our sister office, the Office of Pesticide Programs, or OPPT. And this is a joint draft policy issued by our AA-ship and encompasses both OPP and OPPT. May I have the next slide, please? Thank you. So this is the schematic that's in the policy, and many of you have seen it. It presents the adverse outcome pathway for skin sensitization. Note that the steps along the pathway are labeled, and key events 1 through 4 and non-animal test methods have been developed and accepted through the Organization for Economic Cooperation and Development Test Guidelines Program for the first 3 key events. And Hans Raabe, who Amy had introduced and is from IVS, will go over some of this in greater detail, especially the non-animal methods for key events 1 through 3. May I have the next slide, please? Thanks. So these are the 2 defined approaches that are currently in our draft skin sensitization policy document. And the OECD has a guidance document, number 256, that provides a definition for defined approach. And I like to think of the short version as it's a fixed data interpretation procedure that is applied to data generated from a defined set of information sources in order to come to a prediction or derive a prediction. The use of a defined approach emphasizes that predictions generated by these approaches are rule-based and are not influenced by expert judgment. There have been, I believe, 12 defined approaches for skin sensitization presented as case studies and discussed in various international meetings over the past few years. 2 of these 2 here, were chosen and presented in the 2018 draft EPA policy due to their simplicity and ease of implementation, and we believe that their use will not compromise the protection of public health. So just quickly, in the 2 out of 3, which is on the left-hand part of the slide, this defined approach was initially submitted to the OECD as a case study by BASF, and the approach predicts skin sensitization hazard by testing in an undefined order up to 3 of the non-animal methods that are in the AOP, key events 1 through 3. First, assays are run for 2 of the key events, and if the results are the same or concordant, then the chemical is categorized accordingly, either as positive or negative. If the first two assays provide different results, then a 3rd key event assay is run, and then the overall result is based on the 2 concordant findings, hence the name 2 out of 3. On the right-hand side, we have the sequential testing strategy, and this defined approach was originally developed by Cow, and it's a simple decision tree method that requires tests with either key event 1 or 3. And the way this works is first you do key event 3 assay, such as HCLAT, that's conducted. If the response is positive, you're done, and it's called a sensitizer. If it's negative, you run the in chemical key event assay, DIPRA, and if you get a negative result, it's not a sensitizer. If it's positive, then it's considered a sensitizer. For both of these defined approaches, the only assays that are included are ones with that are OECD test line based and are considered. So that's the brief introduction of the policy. Now I'd like to turn the presentation over to my colleague James Cox, and thank you.
Thank you, Gino. The next 4 slides present an overview of the information that we've received in the TSCA New Chemicals Program on non-animal methods to evaluate skin sensitization. All the submissions, the company names, chemical names, structures, etc., have been claimed as TSCA confidential business information, or CBI. So only non-CBI information is going to be provided in this presentation. OPPT has received 49 submissions with skin sensitization NAMs since the beginning of fiscal year 2016 and up through April of 2020 as part of new chemical submissions. The submissions were received from 24 different companies, and the work was performed by 16 different laboratories, including, in some cases, in-house laboratories from certain submitters. Next slide, please. Here we show some details by fiscal year and the type of information submitted. You'll note that the Lautenberg Act was passed in June of 2016, which was actually the middle of FY 2015. And the skin sensitization interim draft policy was published in April of 2018, which again was the middle of FY 2017. Couple important takeaways from this slide. You'll notice that a little over half of the submissions did not include either an in vivo animal study such as LLNA or guinea pig, or a human repeated insult patch test. That's 27 out of 49, or about 55%. Almost a third submitted both in vivo animal data and NAM data, 14 out of 49, or about 29%. And about the same amount submitted human repeated insult patch test data and NAM data, 13 out of 49, or about 27%. And a handful submitted all three, in vivo animal data, NAM data, and human data, which was 5 out of 49, or just over 10%. Next slide, please. This slide presents, this slide presents whether the submitter used either of the two defined approaches identified in the draft interim skin sensitization policy, the 2 out of 3 approach that Gino mentioned, or the sequential testing strategy method. The results show that the 2 out of 3 method was used in 16% of the submissions, 8 out of 49, and the STS method was not used at all. There was another method used, however, although not identified specifically as a defined approach, which we can term as a weight of evidence analysis. Next slide, please. In this slide, describing the data we received, the submitted information is presented by type of assay performed. We can see that the two most common assays are the direct peptide reactivity activation assay, or DIPRA OECD 442C, which is an in chemico assay, and the keratinocyte response assay, OECD 442D, an in vitro assay. OPPT is evaluating the submitted studies to inform both comparisons and results for TSCA-specific chemicals and to finalize the draft policy. Next slide, please. So we just shared with you the information we've received, and we've started to analyze these data. There are other activities ongoing that will inform the eventual finalization of the draft interim skin sensitization policy. There is ongoing work at NICEDM to evaluate use of skin sensitization NAMs on formulations, mixtures, TSCA chemicals. This last group, work on the TSCA chemicals, is near completion. A case study to develop approach for quantitative risk assessment using the isothiazolinone class of preservatives. This is a collaborative project that's resulted in a recently released document by OPP and uses a different defined approach than what is in the current policy, which is the use of artificial neural networks, or ANN. And finally, comments we've received on the draft interim policy in the summer of 2018. We will evaluate and consider these comments as we develop the final policy. Next slide, please. So this is our final slide, and it shows the formulation for finalizing the OCS PP skin sensitization policy. Consider the comments received from the 2018 draft policy. Evaluate the NICEDM TSCA specific data on skin sensitization NAMs. review of information received in both OPPT and OPP, and recognize and incorporate the appropriate recent OECD activity. Next slide, please. And that wraps up our presentation. I just want to acknowledge Todd Stediford in the Office of Pollution Prevention and Toxics with EPA, and thank him for his contributions. And I believe we're holding questions until the end.
Yes, that's right. Thank you so much, James, and Gino as well. And as James just mentioned, I see some questions coming in. That's fantastic. We will hold all questions to the end after Hans speaks. But if you haven't entered your question already, feel free to do that while the presentation is going on in the GoToWebinar questions panel, and we will ask them after the end of the presentation. So speaking next will be Hans Raabe. He's the Vice President and Chief Operating Officer at the Institute for In Vitro Sciences. He's responsible for the overall operations of the contract laboratory activities and oversees the technical transfer, optimization, and validation of emerging in vitro applications. Prior to his role in testing facilities management, he was the Director of Laboratory Services and Study Director for a wide variety of GLP-compliant in vitro bioassays. His research interests include modeling ocular and dermal irritation and wound healing events in vitro using complex 3D tissue constructs and ex vivo tissues. So Hans, please go ahead.
Great, thank you. And I assume you can see the screen now?
Yes.
Okay, great. So thank you, Christy. Thank you, Amy, for the invitation. I think it's wonderful that we can use these WebExes now to reach out and share on some of our work, particularly in this social distant era we're going through now. So today I'd like to talk about the use of non-animal skin sensitization test methods for a variety of classification purposes, and particularly to support those for the EPA. There we go. So today we'll talk about essentially an introduction to the adverse outcome pathway. I'll go through that relatively quickly. I appreciate many of you have reviewed the background on the AOP development, but I'd like to focus in particular on the various 3 key event test methods, or NAMs, covering key events 1 through 3 in the AOP pathway, and provide some examples and issues as case studies, hypothetical case studies, to illustrate some of the challenges and applications of these methods. Brief to reiterate the comments on the recommended testing strategies and the defined approaches for the EPA, and then close out with some future opportunities, because many of these test methods here really focus on hazard assessments, and I think in the future we do need to move towards formulation risk assessment capabilities. Okay, so to start out, the sensitization events are exhibited through a number of swelling and erythema type events after contact and repeated contacts to certain allergens. And this graphic here comes from Wong et al., who focused on epoxy resin systems as a fairly frequent occupational exposure issue. And they've also listed a number of different allergens and the sources of allergens. And so we can see that a number of allergens come in from the cosmetics and toiletries environment, from personal care and household products environment, jewelry, and so on and so forth, but also for agrochemicals, pesticides, biocides, and industrial chemicals as well. And I think the latter are the areas of focus here for this discussion. So the current in vivo test methods have really focused on the end apical events, the elicitation and outcome of a sensitization event after multiple sensitization induction events, and then the final challenge test to look for sensitization events, and in particular the guinea pig maximization test and the human patch tests really focus on the final apical outcomes. The local lymph node assay has been proposed and used as a more animal-friendly approach by focusing on key event number 4, looking at the proliferation of T cells prior to the downstream inflammation events that may occur after the challenge. So if we think about the sensitization, induction, and elicitation mechanisms, this graphic shows briefly the events that occur with the activation of Langerhans cells after exposure to the haptens or the allergens that may combine with the proteins within the skin cells to form the allergenic drivers. The Langerhans cells bind with these components, and that triggers events in differentiating Langerhans cells and maturing them so that they can do several functions. First, part of the process is to be able to take these allergens and be able to present them to the T cells. The second part, of course, is getting to the T cells, and so the differentiation activities allow these T cells now to, these Langerhans cells to mature and to migrate to the local lymph nodes where they are able to present the antigens to the T cells. This then begins a cascade of priming these T cells to proliferate and recognize these antigens. So in the future, after that sensitization induction step, upon a challenge by that same allergen, these circulating activated memory T cells now are able to effect the inflammatory type responses at the local site of exposures. And if we think about how to take that information and be able to apply that upstream to develop the NAMs and in vitro test methods that have been developed, there are various mechanisms that are very important to consider. And first off, in the exposure to the contact allergen, these contact allergens react with proteins, and they form hapten protein complexes. And those make some notable changes of protein that become recognized as foreign. It's important here in that these proteins and hapten protein complexes elicit some reactions at the local epidermal skin site. In the case of keratinocytes, they recognize those materials as potentially dangerous and irritating. They set off reactive oxygen reactions as well as activate the ARE, antioxidant response element. And so the cells, the keratinocytes, and exposure to these materials release various cytokines. And this is important because those cytokines are also those that now attract the Langerhans cells to the local site where this exposure to this allergen is occurring. So that's a key event that we need to focus on. And then the third key event is, of course, the maturation and differentiation of the Langerhans cells to mature dendritic cells that are able to transform into antigen-presenting cells on their travel to the local lymph node. And we can take advantage of those components there to develop the various test methodologies that are used in the suite of key event assays. So within the skin sensitization adverse outcome pathway, there are various events that occur here. At the final organism response, we've identified the use of the guinea pig maximization test as well as human clinical studies. The local lymph node assay looks at the organ responses, T cell proliferation, as a measure of all the events that presumably occur through this pipeline and cascade to these more or less apical events. We think about these earlier events, the initial interactions with the hapten and the proteins, which is defined as a molecular interaction step. First key event is identified by various test methods. Those in yellow are those methods that are currently adopted by the OECD test guidelines program, direct peptide reactivity assay, the amino acid derivative reactivity assay. For the second key event, where the chemicals and antigens directly affect the local epidermal cells, are represented by the keratinosense assay and the Lusense assay. There are other test methods that are also in the pipeline that look at keratinocyte activation that are not yet adopted by the OECD program. And then downstream, the models for the Langerhans and dendritic cell activation in key event number 3 are identified by the approved methods for the HCLAT assay, which look at the development of CD86 and CD54 surface markers, as well as the USense assay, previously called the MUST assay, which looks at the CD86 marker development, as well as the interleukin 8 luciferase-based assay which is also recognized in the current OECD test guideline. And so for this next series here, I'll focus on essentially the DPRA, the keratin sense assay, and the HCLAT assays as examples of the test methodologies that we're able to utilize to address these three key events and support the EPA program. So the first test method will be focusing on the DPRA, the direct peptide reactivity assay. And this is, as I mentioned, the first key event, the molecular initiating event, which looks at protein reactivity upon contact with the epidermis and the skin cells. And this method has been developed, frankly, by Frank Gerbrek and company at Procter & Gamble, as well as Jean-Pierre Lepoitevin in France at the Laboratoire de Dermatochimie. Variations of this test method have been developed to address various challenges, but this is the basic test method literature here. And the test method basically addresses the process of haptenation, or in other words, covalent binding of low molecular weight substances to skin proteins. Recognize, of course, that lower molecular weight substances are also those that are more likely to permeate through the stratum corneum into the skin and be available for eliciting these type of reactions and responses. And essentially, we've got an allergen. Typically, they're electrophiles that react with, in this case, the thiol group, the cysteine molecule here as a nucleophile, and the interaction produces this allergen protein complex. And that is the antigenic component that drives the downstream activities. The direct peptide reactivity assay looks at the rate of these reactions occurring in chemico or in test tubes. So basically, the assay is conducted by utilizing two different types of synthetic peptides, a synthetic cysteine containing peptide as well as one with lysine. They're prepared at specific molar concentrations in neutral and alkaline pH buffers. Like every assay that's utilized in the NAMS suite, positive and negative controls are included to evaluate the performance of each individual run conducted by the testing laboratory. Test chemicals are mixed with these peptide solutions. Solubility is always a challenge for the test materials. We'll talk about that in a minute. And ultimately, over a 24-hour period after interaction at room temperature, one measures the relative peptide concentration using HPLC with UV detection at 220 nanometers. And so this graphic here simply shows what an unreacted chromatogram looks like for unreacted peptide. This is the target peptide. The second graphic shows the HPLC with the test chemical, and the third one, in ideal situations, will show an evaluation of that peptide and nice separation between the target peptide and the test chemical itself, so that we don't have any interference with the results. In this graphic, one looks at essentially comparing the area under the curve of this peak for the peptide as a control or prior to exposure, and then after the 24-hour exposure to the test chemical. In this case, cinnamic aldehyde as the positive control is used, and shows that the area under the curve has reduced from about 2 million to about 465,000. Then we can take that ratio of that depletion to calculate a percent depletion value for the direct peptide reactivity outcome. And like every test method, one has prediction models. There are two prediction models for the DPRA. The first utilizes evaluation of the mean of both the cysteine and the lysine peptides to calculate whether the material might be a non-sensitizer or a sensitizer. For now, this is going to be, the discussion will just address the hazard potential. So if we have a minimal amount of depletion, some background level of depletion of these peptides, which might be considered minimal, those are really predicted to be non-sensitizers. Anything that's above that level of depletion, above 6%, 6.38% depletion, would be considered a sensitizer. And these are simply looking at the relative reactivities of that chemical at those concentrations with that known quantity of peptide. The second prediction model is used if the lysine data are inconclusive or impacted by perhaps test material interactions or interference at the HPLC. And so this simply looks solely at the cysteine depletion. The vast majority of reactives seem to affect cysteine at a certain level. So again, we have a threshold for non-sensitizer versus sensitizer based on the depletion rate of the cysteine. So some of the limitations and applications of this test method, test chemical should be soluble in an appropriate solvent, and we typically look at testing those materials up to 100 millimolar concentration. In cases where insolubility is an issue, test chemicals can be tested at lower soluble concentrations, but we have to be really careful about looking at those negative results because those negative results may be inconclusive. And this becomes particularly important as we go from simply looking at the ingredient to perhaps working with complex mixtures where the representation of any offending chemicals may actually be quite low and may not be at the maximum concentration tested. There's also limited dynamic information on the rates of kinetic data. In other words, we're simply looking at the total depletion of the peptide after 24 hours, but we don't understand quite the kinetic rate of that interaction with the peptide. But that's something that can be addressed with the kinetic assay, which we'll talk about towards the end of this discussion here. There's also no discrimination of adduct formation from side reactions or information. There can be issues with peptide dimerization, for example, which may potentially give over predictions of the test chemical as the monomer peptides are bound into dimers and are less are then measured as depleted. There's a lack of metabolic activity in this test method, so there's questions on its impact on prohaptin predictions. And since metal compounds, for example, don't interact with proteins by this covalent binding mechanism, but by other mechanisms, this test method wouldn't necessarily identify those chemicals as allergens. So we'll go through a couple of case studies here of the direct peptide reactivity assay. These are hypothetical examples just to illustrate some points here. In this first one, we want to decide whether to use the mean cysteine and lysine peptide depletion prediction model or the cysteine only prediction model. And so in this case, if we look on the far left side here, we've picked up a depletion of this hypothetical test chemical of 12.17% depletion for the cysteine. That is still predictive of a non-sensitizer based on the cysteine prediction alone. And lysine was quite low, very weak response here, clearly non-reactive. But if we look at the mean depletion of the cysteine and lysine, the value of 7.7 is just above that threshold, resulting in a prediction, a positive prediction, if we use the cysteine-lysine combined prediction model. And so we recognize that there's a low cysteine-lysine reaction here, and this would be predicted as a sensitizer. Pretty weak response here, candidly. And so we have several options. We might want to repeat the study to get more clarity. We might use the cysteine-only peptide depletion prediction model, which I think clearly would be under-predicting here, or potentially under-predicting. Perhaps what's also useful in third option would be to look at incorporating other skin sensitization tests. And this really kind of supports the point that in many cases we need to utilize the power of numerous test methods, as was originally proposed in original IATAs, to determine overall whether this positive or negative prediction in this one test method may be supported by other test methods as well. And this really drives the concept of using IATAs and defined approaches. In the second case, solubility is presented as an issue. In an ideal situation, a test chemical has to form a non-viscous solution or non-viscous suspension in any of the preferred solvents for the peptide reactivity assay. And so first, if we have a test chemical that's soluble in appropriate solvent, great. Move forward with the assay, no problem here. But if you've got a non-complete solution and you've got a suspension, if it's not workable, that's a challenge. If you got precipitates, and we'll give some example photos of some challenging solutions and suspensions. Need to inform the sponsor, may very well have to terminate this particular study and try a different approach. But if we do have a workable suspension, certainly want to let the sponsor know this and the primary investigators know this. There may still be a need to terminate the study, but alternatively, we can move forward with the assay. And if we get a positive prediction, that may be a reasonable outcome. If we have a negative prediction, I think we need to be very careful about concluding with a negative prediction. We would typically consider that still inconclusive because it's not clear whether we truly had a negative response or simply lack of availability of the interaction between the peptide and this insoluble material. Similarly, if we have precipitates or biphasic mixtures in a formulation, again, if it's not workable, we may have to terminate the study. And again, if we do proceed with the study, we might be able to rescue the results here with, particularly if you have precipitates forming by centrifuging gently at low speed, and then testing the supernatant in the HPLC and looking for free unbound peptide. This, clearly if we have flocculating of the peptide with the test material, that's very reasonable evidence of reactions with the peptide, and that supernatant would simply support then demonstrating the unbound, unreacted peptide and be able to utilize that in the basis for the predictions. So for data interpretation, again, here's a situation with formulation that was tested with a reaction mixture that formed precipitates. The sample was centrifuged, low speed, and only supernatant was assayed. And in this case, we had very high levels of depletion of that peptide in these two prototypic materials here, both with the cysteine and with the lysine. Essentially, we got fairly high positive predictions here. Clearly, for the hazard assessment, this would be a positive prediction. One can do a bit more to get more information, such as looking at threshold of concentration, for example, to look for a dose effect. One can certainly incorporate other skin sensitization tests, and I think one should, whether you get negative predictions or positive predictions, and certainly use the weight of evidence-based approach to incorporate other information to better understand what might be happening in this case here in terms of this precipitate reaction. Okay, so as we move on to the second test method, the keratinescence assay, this covers the second key event, and this assumes that the allergen makes its way into the epidermal cells and targets the local epidermal keratinocytes. So this assay really looks at the activation of antioxidant electrophile response elements within the keratinocytes. These are stress response mechanisms. And the keratinescence assay, as well as the Lucence assay developed by BASF, both look at this pathway using the repressor protein KEAP1, which reacts with these, which have thiol and sulfhydryl components on the molecule, react with these electrophiles. It forms some changes in that KEAP1 protein that allows the NRF2 molecule to dissociate from the KEAP1 molecule. And the NRF2 translocates to the nucleus and is able to promote and induce the antioxidant response element. And in the case of both the keratinescence and Lucence assays, this activation of this ARE element is based on a reporter construct that also includes the human aldoketoreductase gene upstream of a downstream luciferase gene. And it's that luciferase gene then that gives you a positive response signal that one can measure with the luminometer. And that is demonstrated here graphically. One conducts the assay by seeding these Hackett cells, which are immortalized keratinocyte cells that have been modified with the luciferase gene on the ARE response gene. One seeds those cells into multi-well formats. You treat the test chemicals over numerous test concentrations. In this case, 12 concentrations are used with the keratinescence assay. After a 48-hour exposure, one then uses various luciferase-based reagents. You lyse the tissues or cells and use a luciferase substrate that would allow for the generation of light by the luciferase activity. And one can measure that objectively in a luminescence detector over the whole series of exposure concentrations. And so one generates data. In parallel to that luciferase activity, one also runs the same concentrations using MTT viability assay just simply to assess for cell viability. And so what we're looking for in the prediction of sensitizers is for at least a 1.5-fold gene induction that's statistically significant above that of the control levels. We also need to be ensuring that the same concentrations result in viability greater than 70%. If we have gene induction at lower concentrations or at higher concentrations than are tolerated by the cells, then we may have some false positive responses there. So the prediction model is based on having at least a 70% viability at the comparable doses. And we should see a apparent dose response in this luciferase regulation here. Negative controls or solvent controls are DMSO. Positive control used is cinnamic aldehyde, commonly used in many assays because of its moderate level of sensitization, which allows one to measure increases and decreases in the sensitivity of each individual assay that's conducted. So if we look at the results here, in this curve here, in this first graphic on the left, MTT viability shows that this test chemical here is well tolerated all the way through all the doses, all the way to the maximum dose of 1,000 micromolar. And the gene induction curve shows no changes in that gene induction, and it's well below this threshold line of 1.5-fold induction. So that would be a negative response. In comparison, a positive response would be observed where the MTT viability at levels above this 70% level here throughout the dilution series show a gene induction response. It's both showing a apparent dose response as well as an increase of this gene induction or luciferase generation above the 1.5 threshold. So in this case, we would clearly have a positive response in this range here. In the use of the test method, we should have concordant results from at least 2 independent trials to make a skin sensitization prediction. The viability must be above 70% at the lowest concentration that elicited an induction greater than 1.5-fold. We should see a positive prediction display an EC 1.5 value less than the 1,000 micromolar maximum dose tested. And we'd like to see a clear dose response, but sometimes we see a dose response towards the end that might be covering essentially 1, 2, 3 the doses, and otherwise at lower doses are showing relatively flat responses. And this graphic here simply shows that language here that we'd like to see an induction greater than 1.5-fold. That's statistically significant over the solvent control. If not, negative. If it is demonstrating a positive response, we also want to ensure that the viability is about 70%, and so on and so forth, as we discussed here on the left. So some case studies for the Curatin Sense assays. Ideally, a test article forms a non-viscous solution or homogeneous non-viscous suspension in a preferred solvent. And again, solubility is going to come up frequently in these test methods, particularly the aqueous environment test methods of the cell-based systems. If the test article is soluble in the appropriate solvent, great, continue with the assay. If it's not, you can work with the suspension if that suspension is homogeneous and workable. And this is not unusual for us to see that even with a primary solvent such as a DMSO system, as one moves from the DMSO into the aqueous environment of the culture medium for dosing, one may see some precipitating events. And we need to ensure that we're looking at something that's a homogeneous and workable suspension rather than one that's precipitating out, causing droplets or so on and so forth, which could be an issue for the outcome of the evaluation of the test results. As we go to continue with the assay, we'd like to make sure that we see some evidence of bioavailability, particularly if we get a negative prediction. Do we see cytotoxicity in the response curve of the MTT viability assay? If so, then we're showing evidence of bioavailability and have greater confidence in the negative prediction. So in this case, the test article prediction was positive in the first trial and negative in the second. So what do we do? Well, we have to conduct a third trial, so we have concordant results from at least 2 trials. So a third trial was conducted. In the first 2 trials, we got a sensitizer prediction and a non-sensitizer prediction. In the third trial, we ended up with, again, a prediction that was below that, or showing a EC 1.5 value above 1.5 at below the 1,000 micromolar maximum threshold. And so this would also be predicted as sensitizer. And this is a graphic of the response. So we show viability fully tolerated all the way to the maximum dose. And we see an increase in the dose response here, as well as in the statistically significant increases in the induction values here with EC 1.5 values below the 1,000 micromolar level here. And so this would be predicted as a potential sensitizer. In another example, the test article had an EC 1.5 value that's less than 1,000 micromolar. In the first two trials, an IC30 value of less than 1,000 micromolar. So this IC30 value reflects the evaluation of the requirement for at least a 70% viability by the MTT assay at the relevant doses. And so that is demonstrated here by the MTT viability curve here, showing that at this point we're seeing viability drop below 70%. Since the IC30 value occurred at a concentration less than the EC 1.5 here far to the right, we would identify this material as a non-sensitizer. There is excessive viability impacts or cytotoxicity well prior to seeing any increases in the luciferase signaling here. In a third example, test article crosses the induction cutoff of 1.5 multiple times, as shown in this graphic here. And simply, we want to look at the EC 1.5 that occurs at the lowest concentration, so the first occurrence, to determine if this material is a sensitizer, and also determine whether there's viability here. And so the viability was greater than 2,000, well tolerated, and we would predict this to be a sensitizer, and the EC 1.5 values of 8.3 and 7.1 would be the correct evaluations here. Okay, going up to the third and final test method in this series of 3 key events is one that covers key event number 3 for dendritic cell activation. We'll focus on the H-clad assay. And in the H-clad assay, the test system uses THP1 cells. They're immortalized human monocytic leukemia cell line, and they're used as a surrogate for dendritic cells or for Langerhans cells. These cells have been recognized to demonstrate generation of CD86 and CD54 surface markers as part of the same mechanism that occur with the maturation of Langerhans cells. And so we can measure those induction of CD86 and CD54 markers by flow cytometry. That is essentially conducted by culturing the THP1 cells in multi-well format. You expose the chemicals to the cells to chemical and controls over a 24-hour period. You harvest the cells. These are suspension cultures, so it's easy to collect these samples. You do several wash steps as well as blocking step prior to labeling with FITC labeled anti-CD86 and anti-CD54 antigens, and then you can measure those FITC signals, fluorescent ITC signals, using a flow cytometer. And cell viability is evaluated in parallel using propydium iodide labeling as well. The prediction model requires that if you have a relative frequency induction of the CD86 above 150% above controls, then that would be predictive as positive, as well as a CD54 prediction above or induction above 200%, and that would be the prediction model. And these two articles here talk about the relevance of the exposures to THP1 cells and the use of these cells for modeling the dendritic cell or Langerhans cell predictions. And Galbiati and Corsini had presented this graphic here showing the mechanisms that are involved in the THP1 cells that are mimicking the same events within the cell that ultimately drive the maturation and production of CD86 markers and the ability for these cells, dendritic cells, to be able to migrate to the local lymph nodes. So some of the challenges, again, with the H-clad assay, similar to what we've already discussed with the other methods, are issues of bioavailability, particularly with poorly soluble compounds. And again, we can work with stable suspensions, dispersions, those are acceptable. But the challenges, of course, are with insoluble compounds that produce precipitates or start separating. And there's always a risk of false negatives, particularly with those lipophilic hydrophobic materials with log KLWs above 3.5. This test method really doesn't address any metabolism, so prohaptin predictions are in question. This is isolated outside of the skin in vitro, so one doesn't have the components of any skin metabolism that may be occurring in vivo above the target Langerhans cells, or in this case, the THP1 cells. And something that's brought up by the OECD test guideline, but have at this point not encountered is test chemical fluorescence at the FITC wavelength, but one can readily control and identify that issue even early on as a pre-screen to determine whether that material may even be inappropriate for testing in this test method. And a few H-clad case studies. So the prediction model requires that, again, we have concordant results from 2 runs. And it is possible that we might have just simply in the 2 runs, CD54 positive induction or CD86 positive induction, or both inductions. And those results would be predictive of a positive result and no 3rd run would be required. So if we look at this graphic here, these are all the different combinations for which a positive induction with CD54 and CD86 could occur in both runs, or perhaps one of the inductions, CD54 in both runs, or just the CD86 in both runs. If you have corroborating negative results, you also have a negative prediction and no corroborating 3rd run would be required. But the challenge here is that if we have non-concordant results, where you perhaps would see a positive prediction and negative prediction, or the positive prediction for the CD54 occurred in 1 run and CD86 in the other, then we would have to conduct a 3rd run and then basically evaluate the same type of outcome to determine whether you have a positive or negative prediction. Looks complex, but it's actually quite straightforward. So as we look at a data interpretation case study here, if the test chemical resulted in a positive prediction based on the CD86 RFI in the 1st run and a positive response on the CD54 RFI in the 2nd run, so in the 1st run we got a positive of CD86, but not in the 2nd run. In the 2nd run we just got a CD54. Those would be non-concordant results. And so we really do need to run a 3rd run. And in the 3rd run, the outcome gave us a positive result in the CD54. And so we would conclude that this material was a skin sensitizer. If these both ended up as negative, we would conclude that to be a non-sensitizer, at least based on the results of this test method there. In the 2nd case, the test chemical resulted in a negative response in the 1st run and resulted in a positive response based on CD54 in the 2nd run. And so again, we would run a 3rd run to look for concordant results. In the 3rd run, we didn't get any positive predictions in either of the 2 markers. And so this would be predicted to be a non-sensitizer. So as we look at in conglomeration, all these test methods, they have a fairly wide range of applicability in terms of evaluating chemicals and ingredients. And particularly are heavily used in the cosmetics sector, household products, in industrial chemicals, and as well as in the agrochemical area. But we're focusing with these methods solely on the active ingredients or ingredients within these products. Testing mixtures becomes a bit more challenging. If we test a mixture, higher concentrations and dose ranges may need to be tested to account for low concentrations of any sensitizer ingredients within that complex mixture. If you have a complex mixture with representation of different chemicals at relatively low concentration, we may not be able to account for a positive prediction or have enough sensitivity to pick up if that individual ingredient in that formulation or mixture was indeed a sensitizer. Secondly, some of these mixtures or matrices may actually interfere with the assay itself. So there's a lot of value in testing mixtures using spiked samples to look for recovery of these spiked positive samples in this mixture to make sure that we don't have any interfering events with this complex formulation that might mask a positive prediction. Also, the addition of metabolism and oxidation events to correctly predict prohaptens and prehaptens can be done. In chemical and in vitro assays can include a metabolism component. Using human liver microsomes has been shown to be a useful addition to some of the assays, particularly for chemicals requiring enzymatic activation. The direct peptide reactivity assay has a variant that's called the peroxidase-peroxide reaction PRA assay that allows you to use a peroxidase-peroxide system for certain prehaptens that require oxidation to elicit the allergic response. In the case of the DPRA assay, allow them to be detected. This graphic shows several examples of work that we had done to work with complex formulations to evaluate the impact of those matrices on the ability to detect any positive responses by any active materials within the complex formulation. In the case, for example, this botanical cosmetic ingredient formulation, we're able to utilize the base formulation to spike in known concentrations of known sensitizers and look for recovery of the activity of those sensitizers relative to those same sensitizers tested in a clean aqueous medium and demonstrate whether or not the matrix had an impact on the ability to detect sensitizers. And that gave us then confidence in recognizing whether we could detect sensitizers in these complex formulations. In the case of personal care products, botanical extracts can be quite complex and have concentrations of materials at relatively low concentrations. So we talked a bit about the solubility issues, and this graphic shows some of the issues with various materials here from working with a true solution and a homogeneous suspension. We can see that these are well, obviously the solution is a true solution. This homogeneous suspension is maintained well and doesn't flocculate or precipitate in any certain set time. In this case, we also have a homogeneous suspension, but we recognize that we're losing a lot of material to the glassware and plasticware used to prepare the work. So this is a cautionary issue here. Clearly, with these two cases, we have a biphasic preparation of immiscible liquids and floating particles and precipitating particles here that we clearly don't have any idea whether this material or these materials might be available to the cells or available to the peptide in the DPRA assay. So this kind of gives you a graphic of the range of type of responses we have to pre-screen for to evaluate the test chemical solubility profiles. So we covered the three test key event methodologies here that are utilized in the OECD test guidelines program for in Chemico, for keratinocyte activation, for Langerhans or dendritic cell assays. And the guidance document that Gino and James had earlier talked about, 256, also allows you to report on defined approaches to be used within an IATA for skin sensitization. And the defined approaches really allow one to remove some of the scientific judgment involved and provide very much a defined approach to testing and a defined interpretation protocol to come up with an unambiguous prediction based on utilizing various combinations of these test methods. And so as Gino had mentioned, there were 12 defined approaches that were proposed and evaluated, particularly out of ECVAM, to look at, oops, there we go, to look at their use in various approaches and purposes, particularly for hazard identification, as well as more complex versions for potency prediction. The two that are focused on for relative simplicity were the 2 out of 3 integrated testing strategy proposed by BASF, as well as the sequential testing strategy proposed by CAO, which in the case of the approaches here are really focusing on hazard identification. Nicole Kleinsteuer had done quite a bit of work with Sebastian Hoffman in evaluating the various test methods based on the results of the databases in the Cosmetics Europe database. And whereas this was focused on Cosmetics Europe-based chemistries, please bear in mind that many of the chemicals in here are not solely unique to the cosmetics arena, but are used in multi-purpose functions across a wide range of functional products and ingredients. And so the analyses of the test methods across this chemical space is actually quite useful outside of the cosmetics arena. And one of the findings, I'm not going to apologize for simplifying the next couple slides here. I'll just get to a quick point. I think Nicole does a fantastic job of painting a very detailed picture here, but I'll focus solely here on these results here, evaluating the accuracy of the BASF 2 of 3 approach and the CAO STS for predicting LNA hazards. And they both do a fairly good job, as well as others, in predicting the LNA. And what's important here to remember is that the LNA itself and its ability to reproduce its own results is also about 70 to 80% reproducible for predicting the hazard. So we always have to understand the reproducibility of not only the novel tests that we're trying to evaluate, but also the basis against which we are comparing our results. Also, in comparing the results of these test methods to human data where available, the LNA was able to predict about 74% of the human data based on that limited data set, 128 material data set. The 203 approach and the STS approach did comparable, if not better, in predicting human results, as well as many of the other test methods. And these are the two that we'll focus now on for the EPA program. And so, as Gino had very nicely introduced, the US EPA interim science policy focused on the use of two defined approaches currently for submissions for ingredient materials, the AOP 2 out of 3 and the key event 3.1 STS approach. And we'll touch base on those here in a second. So the AOP 2 of 3 approach for hazard identification allows you to look at any combination of two different key events. So it could be key event 1 and 3, 1 and 2, 2 and 3. And if you get concordant results, you get a positive prediction. And this again is for hazard identification. If they're not concordant, if you have discordant results between the two key events, you then pick an assay from the third key event that would then be used to classify the 203 concordance. And this gives you a lot of flexibility in selecting which test methods, both for timing and other purposes, whether you run them all together, all three, if you want to run 2 out of 3 and go for a third, that gives a lot of flexibility to the investigator to determine which test methods and approaches they would like to take. A little bit more rote is the COW approach that defines that if you start with the 3.1, you start with the HCLAT or the USense or the IL-8 luciferase assay. You start with the key event number 3 test method, and if you get a positive prediction, you have a positive prediction. If you get a negative, you go to the direct peptide reactivity assay or the ADRA, and if you get a positive prediction, you confirm you're positive. If you get a negative in both these test methods, and you have a not classified or negative response. This approach here is prescriptive, but if you get non-corroborating results here, you get a positive, excuse me, a negative and a positive, you may still have the option outside of this approach here to consider what is essentially the 203 approach, and then consider bringing in key event 2 test method to determine if you can get concordant results between the positive or the negative result from the HCLAT or the DPRA assay. So one may have another strategy go back to the 203 approach in practice. I have no idea whether that has been effectively conducted, but that seems a theoretical point that numerous folks have realized. But again, that shows a lot of consistency between the two defined approaches that the EPA has chosen in the program here. Okay, so getting on to the future view. What further info do we need from non-animal test methods? So we looked at this graphic here of the adverse outcome pathway, and we've focused on the key events 1, 2, and 3. And we recognize solubility as an issue. We recognize lack of potency prediction as an issue. So we do want to understand the skin kinetics. How do the materials permeate through to the skin model? Are simply some materials not able to penetrate and thereby not bioavailability in vivo? Can we determine potency for future purposes? And more importantly, can we work with complex formulations and mixtures to move from ingredient testing only to the end use formulation issues that occur in the real world? And can we handle some materials that are above this lipophilic hydrophobic spectrum? And so some of the thoughts here are that there are emerging opportunities for this type of work. For potency, the kinetic DPRA assay allows for potency. This has just recently undergone a ring trial evaluation. The census assay and the EpiSensA assay are both methods that utilize a reconstructed human epidermal model, a skin model, that allow you to measure the material at final formulation in many cases on the top of the tissue and allow for a prediction of both the permeation and the availability as well as the potency of those materials and be able to assign some sort of reactive potency classifications. And then the guard skin also allows one to use a gene expression profile to evaluate potency with more than 200 genes in the prediction model. So the first test method is an in chemico method. It's essentially a derivation of the DPRA assay that is designed to determine the potency of chemicals and be able to assign to GHS classification categories of 1A and 1B. The method is based on a 96-well plate format, so one looks at multiple concentrations of test chemical as well as exposure times. The target substrate or the target peptide is a fluorescent synthetic peptide, cysteine-based peptide, that allows one to look at the depletion of that peptide or the reactions with that peptide as a kinetic measure using a fluorescence microplate reader. And so one moves away from requiring HPLC technology to some more commonly used technologies in many research laboratories. And whereas the standard DPRA assay focuses solely at the reaction after 24-hour exposure, giving you essentially a monotonal response, the kinetic DPRA assay looks at various exposure times. So you can look at the reaction rates across various concentrations, across various exposure times, to look at the rate of which this material may react with the target substrate or the cysteine peptide, and thereby have much more information on the kinetic rates and be able to translate that into potency categories. The genomic allergen rapid detection system, or GARD skin, from Sensitization allows you to use cell-based MUTS 3-cell dendritic surrogates to give you a genomic measure across 200 genes to look for a number of different endpoints, including the ability to see whether the material may be a skin sensitizer. Classification of unknown compounds as sensitizers and non-sensitizers are performed with a support vector machine. So the machine had trained on 38 reference chemicals early on for the development, but this is a model that utilizes additional data in time to improve the predictive ability in the reference database for that prediction model. And Sensitization had worked on a collaboration that allowed for the test method along with a proteomic platform to evaluate this human skin sensitization potential of a glyphosate in commercial agrochemical formulations. And this article had just recently come out just a couple months ago. And then lastly, the platforms I'd like to speak about are both the Census and the EpiSensA platforms. Census is developed by Immunosearch, focuses on 60+ genes that look at irritation and those genes involved in the ARE response, as well as the sensitization responses. And then the EpiSensA assay, which also uses a reconstructed human epidermal model and also looks at gene expression changes, both of which are able to predict skin sensitization of both ingredients and presumably also final formulations. And the beauty here is that one can apply materials onto the top of the skin model in the same manner, same dose and dose rate as one might experience in vivo. So this provides some future opportunities for us to look at potency as well as much more relevant predictions, moving us more towards a risk-based approach. So the test systems are RHE models, reconstructed human epidermis models, commercially available. They include skin kinetics. They minimize solubility issues, particularly as one moves towards testing not just ingredients in various solutions, but also moves towards a concept of testing formulations. The analysis platform is based on gene expression using RT-PCR technologies. In the case of census assay, positive irritation prediction is measured with at least 15 out of 24 skin irritation genes significantly induced. More importantly, for the sensitization response, if 7 out of 17 of the ARE group genes are predicted or induced, or 7 out of the 21, or a combination of both, you would have a prediction of a skin sensitizer, provided that the material is not excessively irritating to the test system. The method is applicable to low solubility compounds. This is fantastic. This is helping us quite a bit, or will help us quite a bit in addressing some of the challenging materials in the more or less dilution-based assays. It's ideal for topical application of complex formulations. Some work has been done by Francois Gautreaux at Immunosearch to work with complex formulations and products on top of the tissue model. It may support predictions using a weight per surface area based data, and this helps us in a more of a QRA approach here in the future, and applicable to mixtures and finished products. And the method is essentially conducted by taking the test system, the three-dimensional tissue construct, and applying test chemical very much in the same kinetics as is used in the skin irritation test defined by the OECD as typically a 15-minute exposure, and then a post-treatment incubation of 6 hours. You have the wash-off step, and you essentially then, after incubation, lyse the tissues, extract the RNA message, and convert to cDNA, and run the PCR on the end samples. And the results are predicted in this manner here to give potency assessments based on the concentrations of positive responses. So a positive response of a formulation or an ingredient at only 50% would be a weak sensitizer. You have more potency, and I'll use that word loosely here. You essentially have reaction rates here in categories that are developed here based on this 5, 10, 1, and 0.1% concentration that give you relative potencies, if you will, to that point that if you have the lowest concentration 0.1 with positive response, that would be considered an extreme sensitizer. And we can relate that then to the activity of known chemicals that fall within these ranges here using reference materials. Okay, so I think that takes me to the end of this presentation. I appreciate the opportunity to speak to you. I appreciate this is a relatively long presentation, covered a lot of ground here, but I think we're then able to open up to questions. And I think I need to then provide this back to the organizers here.
Thanks very much, Hans. We can get started with the Q&A. We have actually quite a few questions, so it should be a very exciting session. I'm going to start with some of the early ones that are directed at EPA first. So in Europe, in clarification data submitted to ECHA, there's been a tendency to have LLNA data submitted when the in vitro data was positive. In other words, maybe they're hoping the LLNA would be negative. Is there such a trend developing in the US submissions as well?
Christy, thanks. This is Gino. We don't know. I mean, we're still analyzing the information. We want to look at each one separately. That's one of the reasons we reported where we have LLNA data and NAM. So we have to look at it more closely.
Right.
That makes sense.
Yep. Another question for you. Will EPA OPPT accept methods that do not currently have OECD test guideline numbers? And how can people know if they will be accepted?
Thanks, Christy. This is Gino again. I'll give a first shot. And as I think many of the listeners may know, when we came out with our strategic plan to address the reduction in testing invertebrates under the Lautenberg Act, we talked about what we considered for relevance and importance for criteria for identifying methods. And we put out an original list in June of 18 and an updated one in December of 19. And we are in the process of developing what we're calling our NAM nomination process. And in the second list, we put non-OECD test guidelines. So with that as a preface, we understand, and from Hans we can see there's a lot of neat things going on that are in various stages of being approved by different parties or entities. But I do want to point out that historically, we've always accepted whatever information we get because we don't have data requirements. So, you know, we'd like to think of the New Chemicals Program as a testing ground for things. And so we do encourage people to submit what they believe is important and information and a justification for why they think it's important. So I hope that's helpful.
Great. Yeah, that actually answers a couple of similar questions. So great. There was a question asking about the correlation between in vitro and in vivo methods, what you presented earlier, and I'm wondering maybe if you're looking into that but don't have that data yet.
We are looking at it. That's one of the important things that we're looking forward to doing. Yeah, I mean, that's why, yes. I do want to specify, this is Gino again, I'm sorry. You know, obviously Nicole, Nicedum, many people have done lots of things in different chemical spaces. What I think is going to be important for us in the TSCA chemical space is, so we now have data on new chemistries, and you know, Nicedum is just about done evaluating 51 different chemicals for which we have LLNA data that were submitted as part of like a Section 8E, and they've done they've overseen the development of KE 1 through 3 for these chemicals. So I'd like to think that this is going to be an important analysis specific to us that'll help inform this whole skin sensitization correlation issue.
Great. So another question is about the evaluation being done by Nicedum that will be applicable to chemical mixtures. They're wondering if this would be for simple or complex mixtures, and what is the status of that evaluation, if you can provide more information.
So this is Gino again. I am less involved in that, and I can't answer specifically. There may be other people who are probably muted that could help, but I'll be happy to see if I can find out more from our OPP colleagues and Nicedum folks. But I don't know the answer.
I think it's just ongoing. Yeah, I don't think I think we'll know when the data is evaluated and made available. Okay, and I think this might be a question for any of the panelists. Some studies have shown the combination of a high performant assay with a less accurate assay. So as is done in defined approach, combining two assays may give a less accurate prediction. What do you think about this hypothesis?
Gino, I don't know if you want to cover that.
Not really. You know much more about it.
So I would say that this is where the weight of evidence really comes into play. I talked about a number of different limitations and issues that come up. In terms of accuracy, we evaluate accuracy based on a public database of chemistries that are, in many cases, fairly consistent across each of the test methods. But indeed, some methods have different purposes and have slightly different impacts and slightly different sensitivities. But I would, I would want to dig into the issues of concern. Why is one method more relevant or less relevant for that chemical space that one's testing, and use a bit more of an IOT approach to understand what's the nature of that chemical, what is its likely mechanism, its metabolites, and so on and so forth, and be able to use that information to paint a clearer picture in a submission to give more clarity and confidence in any of the test methods that are utilized and that have generated data. I think that would be a bit of a weight of evidence exercise there.
Okay. A couple of questions about, I think directed to Hans, about certain kinds of substances. You know, for example, would vegetable oils work as solvents for some of the assays that you presented? You know, what solvent might you use for cinnamaldehyde, which is an oily substance? So maybe some of your thoughts on that.
Okay. In the aqueous dilution-based assays, yeah, we are often challenged with materials that are of high oil-water coefficient values, hydrophobic materials. In each of the assays, we can certainly work with cinnamic aldehyde and other materials in aqueous solution and utilize those, in particular in many cases as positive controls. There may be materials that require initial dilution with DMSO, for example, to solubilize the lipophilic material and create at least a dilution series that is very faithful without having precipitates that occur in the primary solvent, and then be able to move that into the aqueous. But I think what you're referring to there is also, can one use vegetable oil in a test method directly onto the cells? And I would be a little concerned about taking that approach in a relatively lengthy exposure assay, one such as a 48-hour exposure, where we're relying on a lot of ability of the cells to be able to metabolize and maintain normal functions within a medium-based approach. On the other hand, as we move towards some of the three-dimensional tissue construct-based approaches, there a oil-based vehicle may actually be quite useful because that would be well tolerated by the test system by applying that onto the top of the skin model, for example, and would be able to provide perhaps even a very relevant vehicle for presenting a high log KOW material onto the test system.
Okay. And then sort of a follow-on question to that, which is the use of positive controls for the keratinescence assay acceptable, and how does that change the acceptance criteria?
Well, our general philosophy has always been that we need to have positive controls and negative controls with every test system. And the point of that is we don't do it once a month, we don't do it as needed. The point of having good positive control is really to evaluate the performance of each individual trial every time it's conducted to determine whether the sensitivity of the method has been within acceptable control limits, or if the method has become overly sensitive or not sensitive enough, or if some mistakes were made or events occurred that impacted the validity of the study. So you always have to have positive controls in there. And having a good sound database, understanding the upper and lower typical control ranges for positive controls is really paramount in terms of determining whether the results from a test chemical can be reliable. In the case of the keratinescence assay, like every other, we include positive controls with the test system. And we use those controls first off to determine whether the trial is valid. That would be the first thing we'd look at. Was the execution valid or not? If it is valid, then we can move forward to analyzing the predictions of the test chemical.
Okay, another good technical question. What might be the reason that you might see CD54 and CD86 positive and negative with the same test article in the same lab conditions?
I assume that refers to in trial 1 getting a positive prediction and on trial 2 a negative prediction. Mm-hmm. You know, the first thing I would always look at is how close above the threshold are we? Are the results dancing right at the thresholds, or are they strongly positive in one case and strongly negative in the other? In the first example, if it's response is relatively close to the thresholds, those are potentially real events that may occur, and there it would be very useful again to evaluate with a corroborating result to determine the result. Bear in mind that any result that comes close to the threshold is a bit like flipping a quarter, so no matter how many times you run it, no matter how many times you flip a quarter to see if it's heads or tails, you need to recognize that if you're that close to a threshold, the correct answer is you're close to the threshold. In the case of extreme results or extreme differences where you have strong positive in one trial and then strong negatives in the second trial, I would be looking closely at the execution of the work and understanding what might be the result there. And again, starting with the positive control and understanding both from the positive and the negative, what is the confidence in the execution of that assay in the first place? Was there something missed? And again, this gives to the power of then conducting a third trial to determine, and particularly with very different discordant results, determining what might be the correct answer and running a third trial to get corroborating results.
Great. Okay, so we have a couple of questions that are pretty quick about acceptance. So I think these are for EPA. And just to confirm, are these the test methods that were presented EPA approved for pesticide registration data? And I know, Gino and James, that's not your area, but we'll just quickly address that. And then also for TSCA PMN data requirements.
I'm sorry, Chrissy, I didn't understand the question. What was it?
Just asking to confirm, are these test methods that were presented, the NAMs, approved by EPA for TSCA PMN data requirements and for pesticide data requirements? So are they accepted for those two?
Oh, okay, thanks. Yeah, I mean, all the OECD test guidelines, definitely, yes. And that's mentioned in the skin sensitization policy. And as a joint with OPP, I'm pretty safe in saying that that's the case there as well. Does that answer the question?
Yeah, definitely. Thank you.
Okay.
A question about how some of the other defined approaches incorporate in silico predictions. I know that you mentioned the risk assessment that OPP is using or is undertaking that does include some in silico prediction. And so this question is just about plans to increase the use and submission of in silico methods for skin sensitization.
Yeah, this is Gino. I mean, I think that is a reasonable idea. I mean, these were the first two that were identified. We have a couple of years under our belt, and a lot has happened since then. So I think that's going to be part of the discussion to, you know, in terms of finalizing the policy. And we did get a few in silico predictions that came in the door too. Yeah, I agree.
Great. Yeah, I think EPA has had a really open, a nice open approach to encouraging the use of NAMs, which I think is helpful in trying to gain experience with all of these methods. Just a last question for Hans mostly. How to test a product that contains many ingredients for skin sensitization potential? Which ingredients should be tested, or would you test the whole mixture, I guess, or product?
I like that question a lot, and it's not uncommon for that to come up, particularly in more or less a product development cycle, where trying to understand the ingredients as well as the complex formulation in total is important. I think first off, understanding the nature of the ingredients and preparing a formulation with confidence, knowing that the ingredients are either non-sensitizers or formulated at such low concentration that they don't exert a sensitizing effect at the final formulation is a strategy that some companies may want to take. And I would advocate that one understands and tests for the active ingredients that are not known, as well as combine the whole formulation. Also conduct that type of spiking experiment to understand whether that formulation may have a adverse impact on detecting sensitizers by also taking that formulation and spiking in known sensitizers at various concentrations just to demonstrate whether or not one can recover the potency or the activity, if you will, of any spike sensitizers. The challenges are that those get to be pretty large, complex studies, depending on how many unknowns you have. And So budgetary conditions or considerations often prevail in those types of situations, but I would take that multi-phasic approach to evaluating ingredients as well as the formulation.
Great. Okay. Well, we, as always, we have had really a lot of really great questions submitted. I am sorry we can't get to all of them, but we can try to address some of them afterwards. I really appreciate all of you listening, and of course, on behalf of the PETA Science Consortium and PCRM, of course, we really appreciate our presenters, EPA, for being involved and sharing information with us, and Hans as well for going through and really training us in how these methods are conducted and how they can be used. So with that, I think I will close the webinar and hope you all enjoy the rest of your day.
Thank you, Christy.